Many thanks to Hans-Georg for an excellent introduction to what I'm going to say. A lot of experience has been accumulated in the past years, and the first drug I would like to talk about is abacavir. We realized that a lot of patients had hypersensitivity reactions. The genetic test for HLA-B 5709 was tested and approved and developed and very quickly accepted throughout the world. It's very much used. The drug has later got into troubles due to a signal on myocardial infarction. I'm not going into that, but this test in the context with this drug has been very, very successful and is well accepted. I just know of some patients who had been on the drug without having the test made, but the test made after that and had a signal, and they were a little bit worried about continuing on the drug or not. They continued, most of them, I think. The next example is IL-28B polymorphism in hepatitis C, which was already mentioned yesterday. For years, we had been wondering why some patients responded better than other to hepatitis C treatments. Finally, in 2009, 3 publications report that hepatitis C patients with IL-28B CC genotype had spontaneous clearance rate twice as high than IL-28B TT. The association was observed both in white and African Americans. Differences of genotype prevalence in both populations explain the different prognosis in these populations. In addition, the IL-28B CC patients also had sustained viral response rates under therapy with PEG interferon alpha and ribavirin twice as high than the other ones. And the patients with CC genotype had different, the prevalence was different in different populations. So African American had a prevalence of about 25%, Europeans of 68%, East Asians of 90%. So this was very, very interesting, and we learned this, unfortunately, very late. It could have guided treatment responses better at earlier time. Another example are, a less good example actually in the clinic, are genetic factors influencing plasma concentrations when using non-nucleoside retrotranscriptase inhibitors in HIV. We have found some genetic factors. However, these have little impact in clinical practice, as they have shown no explanation for hypersensitivity to nevirapine or to CNS effects by efavirenz. And actually, this is a problem which has affected myself. I have been on efavirenz for many years, suffered of depression and CNS toxicity, wanted to switch to nevirapine, and had a physician who didn't want to do that. In the end, it took me 3 months of heavy discussions with him to make the switch. He was just too concerned due to the hypersensitivity issue linked to Viromun. However, we patients knew that the 80% who actually tolerated the drug did very well in the long run. They had a perfectly safe drug with zero effect on lipids, zero effect on CNS. It was just a pain to start with it. It's very dangerous and very demanding for the physician to start with. And we have never found out, unfortunately, what the association was. We have never found a marker to guide better treatment here. The next topic I would like to come to is tropism testing pre-CCR5 use, which is a drug class also used in HIV. There has been a lot of suspicion among us patients when this drug class came along, because we feared long-term safety issues when using drugs that affect the immune system. The assay, which was around when the drug was approved, has become the cloven foot in CCR5 use. The first assay was done by one lab in the United States. Blood samples had to be shipped on dry ice to the US. and then you waited for 6 weeks to get the results. Now, patients were concerned about this happening because at that time, the United States still had an entry ban for people with HIV, and we didn't know about data protection in the US. There was some concern among us. So nobody wanted to do this test done in the first place. And the physicians didn't want to do it because it was just too cumbersome. Tropism was also very poorly understood at the time when the drug was approved. We didn't really understand if there was switching of tropism or if there was expansion of tropism. We were also concerned about the perceived inaccuracy of the available test. In the long run, it has shown to be very difficult to approve better and more precise assays because the label required the use of the Trophile assay, the first one. However, somehow we managed over the years with Geno2Pheno and also genotypic tests that were developed. However, we run into reimbursement troubles in some countries, and I just did a short check on some European countries. There is no problem in Switzerland at this point. In Italy, the marketing authorization holder has to reimburse the labs. In Germany, the insurers will not pay, but the marketing authorization holder is legally not allowed to pay. Therefore, no drug in Germany at all. In Spain, it depends on hospitals being able to perform the tests. So the picture is a little bit scattery. In the UK, the marketing authorization holder is reimbursing the labs. The assay also impacts the label. In the United States, the drug was approved for first-line use in 2009 on the basis of a retrospective analysis using the newer test, the newer assay, which has shown that the drug was actually safe to use in first line. In Canada, the same happened in 2010, in Australia in 2012. The EMA, however, rejected this documentation in 2009, and in Switzerland, fearing the Swiss regulators would do the same, the marketing authorization holder withdrew an application in 2012. Now, why I am quoting this? This would be, with all we know today, a wonderful drug to use in first line, not for the price, this would have to come down, but for the safety, efficacy, and tolerability, and all these factors are excellent in the long run. This drug is doing better in randomized trials than the placebo. So it's really a great drug for first line use, and it's now currently hardly used where it is supposed to be used. Some areas of improvement where we should work on in the near future. My first case is actually something I'm a little bit worried about because we just had a case in hepatitis C. We had a drug withdrawn in phase 3 some weeks ago, a BMS compound in hepatitis C after 9 hospitalizations and 1 person died. At the moment, industry is very, very nervous when a drug gets to phase 3. People are like that when they see rash hepatotoxicity coming along. People are very worried, and after 5-6 cases, drugs are stopped. And I'm asking myself if we do the right thing. Because I think pharmacogenetic testing should be built as a potential, as an option from the very beginning in these trials. The blood samples should be collected and the trials should run out in the moment when these safety issues occur in order to find out if the drug was safe in specific populations or could be used in the future. And I'm again referring to nevirapine, the case of Viramune. I mean, it's a great drug in 80% of people. It is deadly in 20%. So we need to find out why this happens in order to guide treatment, and we should not throw away drugs in phase 3. We should do that in a phase 2. I know that there are concerns among patients and also among physicians about collecting blood samples for tests to be done eventually. we need some information with the patients enrolled in these trials, and I hope we can overcome this issue. We also need to look at which countries drug samples could be shipped at, because data protection laws are not the same everywhere. I'm more concerned about the United States than about China, actually. Another issue we need to work on, and I can't give you very good examples, but a good scientist told me that this was an issue. Interpretation of pharmacogenetic testing is an issue, especially when there is a binary readout of the data. Then we have regulatory differences across the world. When better assays become available, this should be avoided because patients don't really understand this. My last point is alemtuzumab. This drug has been standard of care for a number of years in patients with a genetic predisposition for chronic lymphatic leukemia. This drug was taken off the market by Sanofi because a lower dose is useful in multiple sclerosis, and the manufacturer wanted to optimize profit. Now, the authorities approved this to happen, and I think industry is really shooting in its legs when something is done like that, and the regulator doesn't help it either, approving this to happen. So this is something I'm not pleased about. In summary, and now I'm dreaming a little bit, and I'm returning to HIV. We have developed about 30 drugs in the past 20 years in 6 different drug classes, if I'm right. We have collected a lot of information about drug-drug interactions within HIV drugs, but also to other drugs. We have collected information about myocardial infarction. We are using Framingham scores in the clinic. We are using all kinds of information, the interactions and all that. We have data about kidney function from the patient, and all this information is scattered out on different websites. So the physician in the clinic has 30 drugs to look at, a patient to look at, and all this information scattered around in different information sources. What I would like to have is either an application on a smartphone or on a tablet with all the drugs listed and all the information from the individual patient, the testing you do per guidelines, coming in to this application, and the drugs showing either red, not to be used, okay to use, yellow, great to use, green. We tried to make this happen in HIV about 4 or 5 years ago. We got the companies interested in this project. We got the money together. We couldn't do it in the end because of IT systems in hospitals throughout Europe being incompatible with such a device. And this is the real tragedy. So I hope we can work on this in the very near future. I also see that payers, and Hans-Georg mentioned to that, sometimes take decisions which we don't understand from a regulatory perspective, decisions we don't understand from a patient's perspective. Sometimes there are contradictions, and it is getting especially worrisome when politicians get in. So this is something we have to overcome, and I still don't know how we can do it. Perhaps we, the patients and the clinicians, can help. Thank you very much.